Well, thank you everybody for coming to the webinar today. My name is Dr. Brian Bauereis. I've been with Azure Biosystems for about 4 years now. Prior to that, I was in academia research working on neurodegenerative diseases, specifically cell replacement therapies. Since leaving academia and joining Azure, I have learned a lot, and it's very interesting to be on the other side of the bench. I've had a lot of reps come and visit me over the years while being in the lab, and now that I get to be on the other side of it, I feel like I've finally come around full circle. So I am going to go ahead and shut off my video now just to increase bandwidth. I just wanted to say hi to everyone so you can put a face with a voice. I will come back on the video towards the question and answer session. Okay, let's get started. So the name of my presentation is Beyond the Blot, Technical Advancements in Imaging and Scanning Technologies. So in this talk, we will explore the technology inside our innovative imaging systems, but also explore unique methodologies that go beyond the blot. Azure Biosystems. We simply do not employ sales professionals. We hire scientists that understand how science and technology go hand in hand. This enables our team to be able to assist with protocols, provide instrument maintenance when necessary, and fully understand complex scientific experimentation to better assist our customers. We work hard in order for you to have the best data to keep your research flowing. We are the only company that does not manipulate data. We are scientists that respect the scientific method and understand what journals require and the trends associated. I will go more into what makes Azure Biosystems the best. We will then move on towards the innovation and the technology aspect. And then after that, we'll go definitely way beyond the blot and kind of give you a sneak peek as to some of the stuff that we'll be seeing in the slides later. I do want to take a quick moment. I am from the New York area. It is almost midnight over here, so I apologize for any troubles that we may have. But also, I understand that there could be language barriers. I will attempt to go as slow as possible. If anybody has any questions or needs me to go slower or faster, just please let me know. So born in California and made in the USA. As you can see here, this is our team. This is not the full team. It does not include everyone. I apologize for those that are missing from that image. But these are the instruments that we primarily work with and sell. This is our Azure imaging system line. This is our Sapphire laser biomolecular scanner. This is our new Azure Cielo qPCR. And of course, we are based in California. Our headquarters is near Silicon Valley, California. Not only is this a geographic fact, but it also should remind everyone that we simply don't follow, but instead we lead with our innovation. First, let me introduce our chief operators here. We've got our CEO and chairman, Eleanor. He comes from an illustrious background where he has been a serial entrepreneur. And then his sidekick, we've got Diping. He has been a successful innovator for many years. If you look at some of the bullet points beneath his name, you might be surprised to see that he was very prominent at Illumina. These are our two chief guys. This is what essentially started the company and what kicks us going still to this day. So a history of imaging. So originally our company was founded in 2013 and based in Dublin, California, of course, near Silicon Valley, where we do design, we develop, and we manufacture all our products. The core members, including Dr. Diping, all come from Alpha Initech, which was then acquired by Cell Biosciences in 2009, and then later Protein Simple in 2011. Back then, Protein Simple wanted to diversify their portfolio, and so less resources went to the development of Western blotting imagers. This prompted Dr. Diping to consider founding a new company where they could develop the best imaging systems and focus solely on such equipment and associated reagents, hence the birth of Azure Biosystems. So we as a company, we do offer a full range of leading Western blotting products to the market. Back in 2014, just a year later after we had founded the company name, we launched the industry's first CCD imager that used lasers and LEDs for quantitative fluorescent and chemiluminescent Westerns. Three years later, we developed the industry's first hybrid CCD imager / laser scanning system that could do phosphor imaging, near infrared, and RGB fluorescent imaging, as well as chemiluminescence. Just a year later, we developed reagents, protocols, and Western blotting accessories to tie in to our two chief instrument lines. A year following that, we developed the Azure imaging system. In 2019, this unit here replaced our C series, so it also got a new name, but the software got a redesign to meet rigorous publication standards of journals while still being the most flexible imager on the market. So as you can see, we do provide a number of these, products and services to get you through the whole workflow of Western blot protocols. I'm not going to stay on this too long because that's not the topic of this, but I just wanted to point out that you can find everything from, gel electrophoresis apparatus to reagents to the high-tech imaging systems. We do have a global sales and support team. I apologize for the people that are not in this. This is just my vanity moment where I get to point out where I live, and currently this is where I'm at right now. So I do service the Northeast sector of the United States. I do work with my colleagues around the country and the world. So let's start in with the imaging technology and take a deep dive as we go on for each slide. I'm first going to focus on the Azure imaging system. This is what used to be called our C series. It did get this redesign. It looks amazing and performs even better. So what does that Azure imager use for its light sources? Well, you may not be surprised to learn that we do have RGB fluorescence, and that is fueled by 3 different LED lights for blue, green, and red. But we also have our 2 laser diodes for the near-infrared fluorescence channels, typically the 700 and 800, plus or minus about 15 nanometers. So excitation light sources. So not all IR is the same, and I'm going to come back to that point because it's rather an important one. So we chose these 2 specific channels of light to put into our instrument because they do pack the most punch in the smallest compact source. So let's first look at LED lights. They're a very broadband light source. They're relatively low power output, very diffuse light, small compact source, and economical price. You can get great data. However, you do have a little bit more background than you might want, which is why we also put in our Azure imaging system, for the ones that prefer the fluorescence, laser diodes. This is a monochromatic light source, has much higher power output. It's a collimated light that delivers more energy to the sample, and it gives you premium performance. So if you look here, if we're looking here, we're right at the tip of that spectrum right there, which means very to no crossover. I was going to say very little to no crossover, with a background that's almost black every single time. Now let's go back to the specific sources of NIR. There was a first generation that used white light with specific filters for NIR excitation. Second generation to follow used LEDs to excite the 700 and 800 channels, improved, but still not optimal. The final next generation and what we chose to put into our instruments are laser diodes. There's less light leakage due to narrow excitation. It's monochromatic and excites closer to the peak excitation, which means you have higher emission signal and best signal to noise. So now we're going to go into the detection technology that we actually use in all of our systems. I implore you to stay with me on this one. I'm going to get a little bit deep into this, but let's see if we can figure out and explain it in great detail. So the first up is the photon multiplier tube, the PMT. It's useful for light detection of very weak signals. It's a photoemissive device in which the absorption of a photon results in the emission of an electron. These detectors work by amplifying the electrons generated by a photocathode exposed to a proton flux, or a photon flux. They are vacuum phototubes that are extremely sensitive detectors of light in the ultraviolet, visible, and near-infrared ranges of the electromagnetic spectrum. Moving on from there, an Avalanche Photodiode is a highly sensitive semiconductor photodiode that exploits the photoelectric effect to convert light into electricity. From a functional standpoint, they can be regarded as the semiconductor analog of photomultipliers. As a photon enters the depletion region and creates a whole electron pair, these charge carriers will be pulled by the very high electric field away from one another. Their velocity will increase to such an extent that when they collide with the lattice, they will create further whole electron pairs and the process will repeat. The avalanche action enables the gain of the diode to be increased to many times, providing a very much greater level of sensitivity, hence an avalanche, like the snowball effect. Moving on from there, the charge coupled device, CCD. It's a device for the movement of electrical charge, usually from within the device to an area where the charge can be manipulated, such as conversion into digital value. This is achieved by shifting the signals between stages within the device one at a time. CCDs move charge between capacitive bins in the device, with the shift allowing for the transfer of charge between bins. Pixel binning is a way to control the gain of the CCD, but not all CCDs are the same. Let's take a dive deeper. So what we're first going to start with, I apologize, a little skip there. This is the charge coupled device. So CCDs allow users to control integration times, choice of long or short exposure, depending on application. And CCDs also capture the entire field of view simultaneously. So the ideal applications for them are high sensitivity with chemi-imaging in long exposures, quick imaging of DNA gels or stained protein gels, or even three-dimensional objects like a small animal or agar plates. More on that later. Back when we go into beyond the blot, I'll discuss those images. Some of the limitations: low quantum efficiency in long wavelengths, limited dynamic range, though much better than film, and then spatial resolution. Let's go a little bit deeper. So this is the CCD technology that I can show you. A lot of this is proprietary. However, this is exactly how they function within the instruments that we employ them in. So there are 5 events that occur. So event 1 is the photoelectric effect. Event 2, they collect the charge. Event 3, they read out, which means they transfer the charge or apply differential voltage across the gates. So essentially, as light's coming in here, they hold the charge, they make it variable, and then this information gets pushed over to the right, where a sequential readout occurs column after column. And then the final event, to detect charge, individual charge packets are converted to an output voltage in digitized form. So now this brings us to dynamic range. Dynamic range is often discussed among sales reps, field application scientists, and individuals just as yourselves. Dynamic range, but what does it really mean? And is there a difference between CCD chips or not? As you can see here, there clearly is. So the size of the CCD, meaning how much electrons can actually hold within that pixel well. Let's say there's 25 electrons or 25,000 electrons with the competitor CCD chip. As you can see with Azzurri's, we actually have 40,000, so it's much larger. Okay, this means that once this gets filled up in the competitor CCD, or if they fill up ours completely as well, it reaches saturation. Larger well capacity means that pixels can capture more light before saturation with the same sensitivity, but with greater dynamic range. If anybody has questions about these, please, as Kai Lang has mentioned, to put them into the comment section, and I'll answer them at the end of the presentation. Some of the advantages of CCD. You might have already figured this out. You get high quantum efficiency. There's low noise, high dynamic range, over 65,000. Some interesting things for astrology. You get a high photometric precision, immediate digital conversion of data. Only low voltages are required. It's rapid clocking and very linear behavior. It's ideal for the work that we put them in on our imaging systems. So this is the Azzurri flexibility and performance. As you can see, this is the Azzurri imaging system. All of these images that you see behind it were captured on that instrument itself. It's rather quite stunning. As you can see, there's some great multiplexing in here. We've got different colored fluorescent, I believe that's bacteria on a plate. We have in vivo imaging of a mouse and a rat. And you can even see IHC brain slices in a zebrafish here. That's phenomenal resolution or a Western blotting imager. Now we're going to switch gears a little bit and we're going to discuss the Sapphire biomolecular imager. This is the first and only laser scanner capable of detecting all methodologies. This includes visible fluorescence, near-infrared fluorescence, CCD-based chemiluminescence, and phosphor imaging. There's no other competition to go against this one. This literally does it all, and it does it very well, as I'm about to show in some of these images. So laser scanner technology. So before we get to the pretty images so that we can all savor with our eyes, let's first discuss the scanner technology. I gave you a brief introduction with the PMT and the ADP detector, but let's go a little bit deeper. Scanners use a scan head containing a point source laser diode coupled either to that photomultiplier tube or Avalanche Photodiode detector. Scan head moves back and forth across the defined field of view, exciting the fluorophore and detecting the emission signal simultaneously. Some ideal applications for scanners: high sensitivity fluorescent imaging, that one seems obvious, qualitative Western blots, wide dynamic range, high resolution scanning, 10 micrometer, tissue section and arrays, and cell culture plate imaging. I just want to take just a brief second to just point out 10 micrometer resolution. I'm not sure the cell types that you guys are researching. I was a neuroscience major myself, and so I know that a lot of neurons are easily within that size, which means you can detect single cells with the Sapphire. That's phenomenal to me. Some of the limitations, however: 3D object imaging, still possible, and I will show you guys how we could actually do that. However, it does have limitations. You can't have massive 3D objects put into the Sapphire, but I'll showcase some of the ideas that we can actually do. Scanning can take longer. Chemi imaging, limited control of exposure time. So now, scanner detector. This is the photon multiplier tube. So the PMT is an electro-optic device that converts light energy into electrical current and amplifies that signal. Gives you great sensitivity due to the internal gain function that helps to amplify weak signals. That's 6 to 7 orders of magnitude. Ideal for high sensitivity fluorescent detection in RGB, and diminished quantum efficiency at wavelengths of greater than 600 nanometers. So that means maybe this isn't the greatest idea that we could use because it has that diminished QE, but we're going to touch on that in just a second. Second, the Avalanche Photodiode. It's that highly sensitive semiconductor that exploits that photoelectric effect to convert the signal to current, provide a built-in first stage of gain through an avalanche multiplication, similar to PMT. APDs have high quantum efficiencies across the spectrum, especially at longer wavelengths, greater than 600 nanometers. So we sought to identify which detectors would be best for the researchers that is conducting the science, not what is going to be the cheapest, not what's going to just give data. We sought what's going to be best. So we used the PMT for quantum efficiencies lower than 600, so just in the RGB range. And then for near infrared, we used the Avalanche Photodiode. And that's very evident if you look here at this graph here. On the y-axis, we have quantum efficiency. On the x, we have the wavelength. As you can see, PMT, it's quite low and drops rapidly as you gain in wavelength. So we get further out to that near infrared, it drops significantly. Whereas the Avalanche Photodiode works excellently. So if we're talking 800 nanometers is about the max that we would do for near infrared. And you can see it's, leaps and bounds above the PMT right here. So obviously this was a great choice for us for the near infrared spectrum. So now we are, basically this summarizes what we just talked about. So we have the photon multiplier tube. It's highly sensitive photodetector in RGB. And then we have the Avalanche Photodiode, high quantum efficiency in the NIR. Best of both worlds. I'm not sure why other companies don't employ the same technology, but we have it here and we hope that you take advantage of it. So dynamic range, it's dependent on the detector technology. So why dynamic range is the ability to capture both very low and high signal intensity in the same image without saturation. As you can see, film, we go back in the day, although there are still some classical scientists and even newer individuals that still use film. I understand that, but hopefully, as you can see, the dynamic range goes up significantly as we move forward over to the CCD device, the PMT, and then the Avalanche Photodiode. Every detector has an intrinsic dynamic range. PMT and APD have the widest of them. So now we're going to get into probably what you guys were hoping to see, and this is some of the more applications that go beyond the Blot. And most of them we're going to focus on the Sapphire. However, I do believe that we will discuss some of them that have the Azure imaging system used as the source. So let's first go into proteomics. Proteomics is probably one of the most rapidly studied fields in science and medicine, specifically here in the USA. You know, there's roughly 20,000 human genes, which means for proteins that we have either 20,000 proteins that go all the way up to 500,000 proteins. So it's clearly the higher spatial resolution data set that you want to go towards if you want to find every subtle intricacy that exists within a cell that you're studying. So while 1D polyacrylamide gel electrophoresis is great for most applications, many proteomics and other studies do benefit from an additional dimension of separation to resolve co-migrating proteins and their isoforms. So you can see here that we show it's a close-up of a 2D protein gel that was stained with cyperruby. With a 50 micrometer resolution scan, you can easily see the distinct spots, which can also be quantified in the Azure Spot software. So this is 50 micrometer. It does go all the way down to 10 micrometer. I mean, that's excellent resolution. So we're going to see better in future slides. For more sensitive detection, 2G gels can be run with protein sampled isolated from cells grown in the presence of 35S labeled methionine. The radio label becomes incorporated into the cellular proteins, which can be directly detected using the Sapphire's phosphor imaging capabilities. So as I mentioned before, the Sapphire, it does everything to include chemiluminescence and phosphor imaging. So if you want to take a step further with your research, the Sapphire can do it. Another interesting approach in science is the electroparetic mobility shift assay. This assay, also known as the gel shift assay, if you haven't heard the EMSA before, it's a great way to monitor any type of stable binding reaction, such as protein to protein, protein to ligand, or protein to DNA. This technique can be used to analyze sequence-specific interactions as complexes of protein or protein and DNA migrate slower than unbound protein or DNA, causing a shift in the bands within the sample. Traditionally, these EMSAs are performed with radioactive isotopes, but the technique can also be adapted to use like non-hazardous fluorescent dyes, which can decrease assay time by cutting the time required for film or screen exposure. EMSAs provide you with the ability to measure protein DNA. So here we show the results of testing the Sapphire's ability to image a protein DNA binding reaction using EMSA. DNA is shown in red and protein is in green. Okay. The powerful lasers used in the Sapphire enable imaging of the gel directly within the glass plates, reducing the risk of breaking these delicate gels during the transfer to the blotting paper or while drying. And then, of course, the measurement of bound and unbound DNA is easily accomplished in the Azure Spot software. So these are representative images of what the Azure Spot software kind of looks like as you go through the steps. So you can definitely identify the bands by looking at the lanes. You can get all sorts of histogram graphs to look at your data. And then, of course, you can even calibrate the volume of each individual lane. One special researcher is using tracking of movement with the Sapphire, and they are starting to get some tremendous results. So here we show that the fluorescently labeled antigen, it was injected subcutaneously into the mouse hind paw. The animal was euthanized, and fluorescence from the draining polyteal, or the back of the knee, and sciatic lymph node were measured. Numbered green circles indicate areas where the signal was to be measured. So as you can see here, we are looking at three-dimensional objects. We are looking at them in a 2D representation currently. However, I do want to tease you that we do have some 3D capabilities coming up. Some further work to track protein movement was done with another example of looking at protein distribution in different tissues. So this experiment And I apologize that the data isn't as clear as I would like it. This was some initial data that I received from one of my researchers in New York City. But it does show the administration of an IR dye conjugated to an antibody that cannot cross the placental barrier. So embryo on the right versus conjugation to an antibody that can't cross the placental barrier, embryo on the left. By imaging with the Sapphire, rather than with a camera and filter setup, you can quickly observe protein localization across distal tissues and easily quantify relative protein distribution. So no longer are people buying these instruments just for simple Western blotting techniques. People are definitely getting more out of the blocks use with the instrumentation due to the great optics, detectors, and all the technology that we pack into the Sapphire and Azure imaging system. To further look at this, there's another researcher that sought to track the viral load and quantify that amount in zebrafish. So the Sapphire can be used to track localization of more than just protein. So in this demo, FITC labeled virus is used to infect a zebrafish, which is then placed directly on the Sapphire for imaging. And if it wasn't clear, this is the glass plate of the Sapphire. You can actually see some of the cameras and laser detectors right beneath it. And look how gorgeous that is. That's beautiful. Now, one of my favorites, clarity. So it was originally developed to help neuroscientists better image entire brains. The clarity technique is a way to optically clear brain tissue while preserving biologically important molecules like protein and DNA in the context of larger brain structures. In a manner similar to fossilization, lipid bilayers are replaced by a sturdier yet porous and clear hydrogel mesh. Labeled macromolecules lying deeper within the brain can now be imaged. With the flexibility and powerful focusing power of the Sapphire, you can obtain wide field imaging of clarity prepared brains from small animal models. So I did take these two images from the web. They were not taken on the Sapphire, but I want to give you a brief introduction to what clarity is and the powerful nature and beauty that you can actually uncover. So yeah, just imagine removing all the lipid bilayers of the brain, replacing that with a hydrogel mesh, and now you can actually fluorescently label individual neurons, even as groups. You could do glial cells, astrocytes, pyramidal neurons, and give them all a distinct color. And as you move in through the tissue, with special microscopes, mind you, you would not be able to do exactly going into the tissue with the Sapphire. We can do a kind of Z stack in which we go up and down the Z range by a few millimeters at a time. I do have images for that, and I will get to those shortly. So clarity does have some other interesting aspects to it. And so clarity brains, like the ones that I have imaged here, this was taken directly from Dr. Cal. She's worked at the Feinstein Medical Research Institute in New York. And she provided me with this first image that she had taken of the clarity brain that they had prepped there. Again, I have to apologize. She doesn't have all the data and is not prepared to send it to me just yet, but they have yet to actually go into the brain. They just sought the protocol to make sure that they could actually get rid of those lipid bilayers and make the protocol work. Apparently, it's not an easy one to accomplish. Nevertheless, she got the first brain to work, and now she just needs to radio label it or fluorescently attach antibodies to it to actually define specific structures. So now cell-based assays. There's obviously high throughput protein detection as probably the most important one. And so while Western blotting has been the lab standard for decades, the high performance of the Sapphire enables time and labor saving extensions of the Western blot that would have been hard to imagine when the technique was introduced. One such extension is the in-cell Western blot, where plate-grown cells are fixed, permeabilized, and then probed with antibodies in situ. The result is accurate measurement of the intracellular protein expression while the cells are still in the plate, which provides a high throughput method for assessing multiple stimulations, endpoints, proteins of interest, and replicates on a single plate. By using the NIR antibodies in the Sapphire system, the potential for in-well multiplex analysis also exists, offering further improvements to the throughput. This is a great. The in-cell Westerns can absolutely save you a ton of time. If you just simply look at the steps that we've put here to the side of it, you literally do everything within the plates themselves. Standard imaging systems, you would not have the resolution to see this. However, with the Sapphire and the 10 micrometer resolution you can achieve, you can actually get protein localization seen in between each well, and you can quantify that. I had to throw this in there because I thought it was funny that we're discussing everything that the Sapphire can do outside of the Blot. Of course, it can do beautiful fluorescent multiplex westerns. I'm not going to leave this slide up long because it's not that interesting, but it certainly does that, and it does it with precision. So imaging possibilities for gels. What can you do with a gel on the Sapphire? Well, all this stuff. Proteins with TAMR or GFP. You can look at protein native gels. You can even look at RNA gels with Nancy dye, or even quantum dots in gels. All of that stuff is achievable on the Sapphire. Moreover, if you're interested in doing fluorescent microarrays with the 10 micrometer resolution, it looks absolutely stunning. You can see the individual dots, you can see them in great detail. Now we're going to get over to the little more visually stimulating section of the presentation. So a lot of the images were obtained from my colleague, Dr. Andreas Schroeder. He's, I believe, or Nikki might have to correct me, but I believe he's over in Switzerland. Nevertheless, as you can see, the mouse brain on the left and Drosophila melanogaster on the right. I mean, look at that. If you're needing to identify specific structures anatomically or even just the morphology, you can use a Western blotting imager. You can use the Sapphire system. Here's another great one, mouse hind paw, again by Dr. Andreas. That's just gorgeous detail. Again, if anybody has any questions on how these images were obtained, speak up. If you want to provide us with your email address, we can send you specific protocols. This is one of my favorite ones. Look at that anatomical structure. We're looking at mouse embryos. You can see every one of these tissues in it. They look absolutely stunning. You can even make out some of the small, let's see, yep, there we go, right in this. You can see the spinal cord right there, each individual vertebra. So you can actually watch things develop with the Sapphire system. And look at this. This is a whole mouse scan. How beautiful is that? So if you're needing to get any sort of holistic view of your animal, if you're looking at disease modeling of something that's as small as a single protein, obviously you can do that with a Western blot, but if you want to take it back and do a whole scan, that's where the Sapphire shines. That's so gorgeous. This is mouse lung. The preparation is very simple. And look how gorgeous that looks. Just by separating out the distinct colors by using the different lasers, you get a beautiful image like that. This might get you guys published on the cover of Nature. How about this kidney? You can actually see the individual tracks within it and the individual cells as they radiate away out of this pathway there. That's stunning. 10 micrometer resolution. How about this? This is cow stomach lining. Beautiful. IHC rat brain. So immunohistochemistry of a rat brain. It's not as fine as detail as a microscope, so I always recommend to my Sapphire users to image all their slides on the Sapphire for visual documentation of their IHC experiments. I never had that before. I used to have to write down, as you can see here on this glass slide, all this information that you want to have packed in there. Now with the Sapphire, you can just line up your glass slides and take a whole image of all of them together, and there's your visual documentation to put in your notebook. Now, I did tease you guys with a little bit of an intro to a Z-stack that the Sapphire can accomplish. So what we're looking at here is a grasshopper, again, Dr. Andreas, and he took these measurements below, the 0 to 0.34, 0.66. He's backing away. Those are micrometers. And you can see the clear level of detail on each scan get you a little bit more or different information that you had prior. Hopefully you guys can see this okay. Right now we should be seeing a GIF of the grasshopper going in and out of that pseudo Z-stack. How gorgeous is that? And if we zoom in a little bit closer, he's looking at you guys. That's just absolutely stunning. This again is a Western blotting imager. So just the power that we packed in with the technology, it's phenomenal. And I implore you guys to consider it in your research. Some more imaging possibilities for blots. You've got the 2D blots. You have plates, colonies of E. coli in blue light, colonies in white light. We also have E. coli, plate GFP, and M. cherry, as well as, I did have this image earlier in the slide. Of course, that is E. coli, GFP, and M. cherry. Plants, if you're doing botany research. Imaging possibilities for animal samples. Again, we have fixed mouse brain slides and even egg embryos in white light and their bioluminescence. There are possibilities for in vivo work as well. Obviously, we just have a regular mouse in white light. Luciferase in vivo bioluminescence. We have an in vivo mouse doing RFP detection. They were specifically looking at a GI tumor, I believe. Zebrafish, you can see the expression in vivo. Chicken embryos, with or without the virus load. This is the very interesting Newcastle disease virus that actually caused substantial economic losses to farmers in, I believe it's low and middle income countries, with high levels of morbidity and mortality in poultry flocks. So a very interesting use of the Sapphire, and it worked beautifully. Of course, in vivo RFP detection, I did blow this image a little bit more so you guys can see the great detail. Obviously, some preparation has to occur on the animal. If you guys have questions about that, we'd be happy to provide protocols. Of course, mice and rats, they do autofluoresce their fur, so some prep has to be done. Some more possibilities in vivo. Look at this. We have internal liver tumor, so RFP in vivo. Another liver tumor, GFP. Subcutaneous tumor, RFP, and the subcutaneous tumor, GFP in vivo. How great is that? So the Sapphire lasers do actually penetrate the dermal tissue. I believe it's about 5 millimeters. If I am wrong, I would apologize and send you guys the correct information. But I believe it's 5 to 7 millimeters. So you can actually see stuff that's inside the animal if you do fluorescently label it. Here's another subcutaneous tumor, blown up image. And look at that resolution is absolutely stunning. You can actually see the individual hairs of that animal. So what does this all mean? This was pretty awesome, I must say, which is the whole reason why I left academia. This company, I do akin it to Apple. I apologize if you guys are not Apple products, but you know, they're also based in the Silicon Valley area. They make great products that are easy to use, that are powerful, intuitive. We do the same stuff. We're very proud of it. As you can see, we do cover the entire workflow. So as you go up in performance and up in price, you can see that we do have more and more powerful imaging systems. We do sell, of course, all the reagents. We do have basic gel imaging, which is our C200 model. It doesn't have all the fluorescence if you don't need it. So we do provide very powerful systems that don't have all the bells and whistles if you don't need them, but they are upgradable in the field. So as I'm approaching the conclusion here, I did want to point out that Dr. Andreas, you know, he did submit this image to WikiScience competition, and he in fact won. This is a beautiful honeymelon image that was taken on the Azure Sapphire imager, and look at that. It's absolutely stunning, 10 micrometer resolution. Just imagine any of your samples of science put onto the Sapphire, you would see a gorgeous image like that. It's amazing. This is the end. I thought this was a funny conclusion. You know, ever make mistakes in the lab? I certainly have. And look at this Western blot. It doesn't look that pretty. Let's make them birds. Yeah, they're birds now. Thank you so much, everyone.